be all of these little blips in here. Okay, those are spines where the site for excitatory synapses. And he realized that pyramidal neuron like this one, it will have like about on the order of 1,000 and 10,000 spines. So if you can regulate the strength of each one of those spines, or each one of those synapses, that would be an excellent mechanism for coding new memories, because you will have an immense buffer capacity. Okay, so if each one of the 10,000 synapses can have, like, say, 10 states or 100 states, then you can have a buffer capacity of very, very big size, just by that. Okay. Another possibility that people thought it was also you can also code memories. You can change the properties of the network by extending or retracting dendrites with all of the synapses in there. Actually, that happened. So sometimes after many presentation of something, you can get a retraction or a prolongation of dendrites with all of the synapses in there, but the limitation of that mechanism is very slow. So it will take several minutes or on the order of hours to make a reasonable growth, and memories can form in seconds. So it doesn't match. But changes in the synaptic strength, that can be fast, and that's what we're gonna concentrate. And the idea, and in here we will see many instances in which a theory precedes the experimental. Okay, so all of the idea that we have now about synaptic plasticity and memory was foreseen by this guy Donald Hebb, and he thought a very simple way of visualizing memory formation. It was the following. He said, suppose that we have a very simple system, in this case with 10 neurons or something like that, interconnected. Okay? Then you present a stimuli, which is this annulus, and then you activate those neurons. And since they are interconnected, they tend to be active for a little bit longer. So this one activates this one, that one, and then you can get kind of like a reverberating activity for a while. And then he thought, well, if there is a process during this reverberation such that cells that are firing at the same time the connection gets stronger, okay, maybe you have a situation in which some of these connections are stronger, and that could be a way of coding the memory. And to test that in this conceptual experiment, it was like, well, what happens if we present a partial view of the initial stimuli, okay? And so the annulus, these pieces here. Then the interconnected neurons will activate themselves, and then you will get basically the same ensemble of neurons firing again. So that property is called pattern completion, and it's an essential aspect of memory formation, basically. Okay, so you should be able, if you have a proper memory, you should be able to recall the memory just with partial information. Okay, and a simple network could do it, that in principle. Okay, so
how does this relate to sparsity, if it does at all?
That's not related to sparsity. Sparsity is another aspect of that. You want it to be sparse, so you can imprint different memories in the same network without interfering. Okay, that's there. And there's another phenomenon called pattern separation that you want to do. We're not going to talk about that, okay, in here, because we're going to talk about the basic mechanisms at the synaptic level. So what happened? So Hebb postulates a very famous postulate now, so that here, when an axon A is fired and it makes the axon B to fire, some process must happen. He didn't know what it was at that time, such that next time cell A fire, the efficacy of making the cell B to fire, it will increase. Okay, so that's called Hebbian association, and actually we will see that actually happen. Okay. Interestingly, that was done in 1949. It took more or less like 20 years to somebody to thought a complementary. Kind of like, oh. So if cell A failed to make cell B to fire, or cell B fire for other reasons, then that connection should be weakened and reduced. Okay, so that would be the two complements. Okay, and both of them, we will see both of them happen, and pretty much on the same, in the way that it was envisioned. Okay, so I'm gonna talk here. The Hebbian changes are called LTP, long-term potentiation, and the anti-Hebbe changes are called long-term depression. Okay, and I'm gonna talk about the induction, expression, and maintenance. So the induction, it will be, well, what are the actual neural signals that trigger potentiation and depression of synapses? Expression, it will be, which are the actual changes at the synapse that make the synapse stronger or weaker? And maintenance, which is actually kind of like the interesting question that's now, how you can make those changes to persist during the lifetime of a individual, okay? And that's very important because synapses are subjected to molecular turnover. So eventually, within a month, every single molecule that you have in a synapse has been degraded and replaced by another one. So you cannot code the memory in the synapse just simply by modifying those molecules, because those molecules are gonna go, okay? So it must be a system that preserves memory. So how memory can be preserved in a synapse, that's it. And the answer is nobody knows. So this graphing here is just to show you the interest of long-term potentiation or long-term depression. What I did here is I plot the year and the number of papers. Here is the first, in 1973 was the first demonstration that synapse can change. And that's 19, okay? And then for many years, for almost like a decade, nothing happened, until you will see somebody invented, I'm going to show in the next slide, the slice preparation, that you can study synapses in a much easier way. And then there was like an exponential growth on the number of papers. I entered the field about here, so that was a wrong thing because it was about saturated. But anyway, but you can see now that you have like about 1,000 papers published per year, and probably by now, this is an older figure, probably like 2,000. So you're going to get a very simplified version of the field of long-term potentiation. Okay. So at this very moment, while I'm talking, probably there are 2 or 3 papers popping out on the internet about long-term potentiation. So I'm going to compress all of that in 40 minutes. Okay. So this is the basic preparation that I'm going to refer is the hippocampus. Okay, so the hippocampus is very important for learning and memory. Probably many of you are familiar with the HM. If you lost the hippocampus, you cannot acquire new memories. That's put it like shortly. And in the rat and in the mouse, the hippocampus is like a little banana that goes like this. Okay, on both sides. And the cool thing about this banana is that you can chop it like a salami. Those are slices, okay? And each one of those slices preserve the fundamental connectivity, okay? So this will be, let me show you, this is the banana, and this is one of the slices of the banana. And basically, information activity enters the hippocampus through here, the perforant path, go into these cells, the granule cells. From these cells, go to these other cells, CA3 cells, and from CA3 cells, it goes to CA1 cells, and then it gets out. Okay. Actually, there are more cells in here. Between CA3 and CA1, of course, there is CA2, all right, and then there is a CA4, and there's all kind of, but other ones. But these 3 are the most, people would say, the most relevant and the most important. And all of these 3 synapses show plasticity. Okay, but we're going to concentrate on this synapses from CA3 to CA1. And the reason is totally pragmatic. Okay, that synapse is the gift of nature to neuroscience. Okay, so there are several things that are good about that. First of all of the axons from CA3 goes into CA1, and there's nothing else in here. So basically, you put an electrode here to stimulate, you're pretty much sure that the only thing that you're stimulating are those axons. And if you put an electrode in here to record, you're pretty much sure the only thing they're recording is the CA1 cell. So it's simple. It cannot be more simple in the whole central nervous system of a vertebrate. Okay, that's simplest. And it's very resilient. The same joke I tell every year because I work with these things. If you are walking and you drop the slice on the floor, you just don't tell anybody, you pick it up and keep working, okay? And it's very robust. If you're doing the same thing with a slice from the cerebellum or the cortex, actually, if you look too strongly to the slice, you can kill it, okay? So it's very resilient and very easy to interpret. It's a pleasure to work with this thing, okay? So people work with that. And what they do, okay, so here's the experiment. So you put a stimulating electrode in here, which is basically a rod, two rod to pass current, and you record here. Normally you can record either extracellularly, current going into the cells, or you can poke a cell and record intracellularly. And in both cases, you record a response and you can quantify it. And here you have time versus the response. It's called excitatory postsynaptic potential. They use the slope of the response for some reason that I could explain you if somebody asked me. If not, I will not explain. Okay? So those responses are very stable. Okay? So it's about 100%. And at this point, and you stimulate normally at 1 or 2 per minute. At this moment, what they did is that they give a high-frequency stimulation. Instead of 1 per minute, they give 100 hertz. And it was really like, oh! All right? And you get a big potentiation, and then actually, after you resume the normal stimulation, the things remain high. And in this case, they repeated 4 times this tetanus, and the whole thing remained high for about 3 hours. That's about how long you can keep those slices alive. So actually, it's really long-term potentiation. So the response gets long-term. And memories can be long-term. I mean, if you have a traumatic memory, you can the memory form in 1 second, and then you can remember for the rest of your life. Okay. The record of duration of LTP is held in New Zealand, in Otago, by this guy, Cliff Abraham. So he do the same experiment, but with a live rat. He put a stimulating electrode and put the recording electrode, seal it, okay, and record it for several months. So he made the LTP, he gives the tetanus, and then record how long the responses remain stronger, and actually it was 1 year. So the Guinness record of long-term potentiation is 1 year so far in a rat. So basically, those synapses remember for a year that for a few seconds they have a high-frequency stimulation, and that's kind of like the essential property of long-term potentiation that make it so attractive as a mechanism of memory, because you can induce it very quickly, and it can last very long. There's several order of magnitude difference between how long it takes to induce and how long it can last. Okay. And here I'm going to go through a few of the important properties of long-term potentiation that make it so interesting. So first of all is input specificity. I told you that you have 1,000 synapses, and you want all of them to be independently modulated. If all of them, if you induce LTP, but you're inducing all of them at the same time, then there is no buffer capacity, basically. All of them, you can collapse it into one. So you have to be independent. So to show that they can be independent, what people did is they put 2 stimulating electrodes in this part, and they record in CA3 cells here the responses. Okay, and what they did is that they applied the tetanus, the 100 hertz, only to one pathway, pathway 1. Okay, and that pathway, that was before the tetanus, and this is like, say, 1 hour after the tetanus, it gets much bigger. But the other pathway that was not touched during that time, that was not tetanized, it did not change. So only synapses that are conditioned with the tetanus change. The ones who are not do not change. So that's input specificity. The other thing that people notice is that you need a minimal amount of axon to be recruited simultaneously to produce long-term potentiation. Okay? So you can adjust the intensity of the stimulation to just recruit one axon, for instance, to recruit, say, that axon. If you do that and you give the tetanus to only that axon, nothing happens. Okay? So you need to co-activate a certain number of axons together. And actually, the number for the rat, at least, it was 8 axons. So you need to co-activate 8 axons together in order to get RTP. Fewer, it will not work. All right? And here is the last property, which is very beautiful, which is associativity, which is kind of a combination of cooperativity and then input specificity. So what we have here is two pathways, and this pathway we stimulate not with a tetanus, but a low frequency. So normally you stimulate to test 1 per minute, but here let's stimulate at, let's say, 1 hertz. It's not that strong, but nothing happened. Okay. Here we have another pathway that we can give the tetanus 100 hertz. Okay. So now if we co-activate both of them at the same time, so while we're giving 100 hertz to this pathway, we're giving the low 1 hertz to this other pathway, it turned out that the other pathway, the weak pathway, so to speak, the low frequency pathway, get also potentiated. Okay. Somehow this pathway help this weaker pathway to get potentiated. And this is associative, and it's basically very beautiful because it's a synaptic model of Pavlovian conditioning. It captured the essence of Pavlovian learning. So because you can remember in the Pavlovian experiment, you take the dog and you show them the unconditioned stimuli, which was the steak, and the dog salivate, and then associate the steak with the bell, which is nothing happened normally, but if you associate them, then the dog will salivate with the bell. So this pathway now is the steak, the strong pathway that makes you salivate, and this pathway is the bell, the weak pathway that normally doesn't make you salivate. If you associate both of them, now the weak pathway, it will produce potentiation. Okay, so so how it happened? So far we know is the NMDA receptor. I think you're familiar with the NMDA receptor, are you? Huh? Yes or no? No? Oh, who say no? Okay. All right. Okay, so this is a cartoon of a synapse, a glutamatergic synapse, and normally they contain many receptors, but these are the two more important. The AMPA receptors that are the one that once the AMPA open, allow the cations go back and forth. So this basically responsible for the depolarization of the cell, and the NMDA receptors. The NMDA receptors also allow cations, but also importantly allow calcium to get through it. Okay, but normally the NMDA receptor, even if it binds a glutamate, okay, here's the glutamate coming from the presynaptic terminal. Even if you bind the glutamate, nothing happens because it's clogged by a magnesium ion. Okay, so the only way for the NMDA receptor to be fully activated is that if this postsynaptic cell is very positive, very depolarized, and in that case, the magnesium get out, and then this guy can conduct freely. Okay, and so therefore, for that reason, it's called the coincidence detector, because it detects the simultaneous activity of presynaptic and postsynaptic. Presynaptic is because you need the presynaptic to release glutamate. Okay, so the NMDA detects presynaptic activity, and it also detects postsynaptic activity through the magnesium binding. Okay, so when you have the coincidence of pre and post together, then it's when you can fully activate the NMDA receptor. And actually, that is the HEP rule. HEP says when cell A makes cell B to fire action potentials, then you will initiate a process to potentiate that connection. So the guy who's reading that those circumstances basically is the NMDA receptor. Okay, so it's the molecule of plasticity or whatever. Okay, so the way that people show that, one of the way to show that is that in order to fully activate the NMDA receptor now, what you need is you have the NMDA receptor, you bind glutamate, and then there's also the magnesium block. So you need the postsynaptic depolarization to release the magnesium block. And actually, that's why you need the tetanus. Okay? So what happened during the tetanus is the first response is like this, and then the second response is like this. And so during a high-frequency stimulation, basically you are piling up responses, so to speak, and on a voltage. You probably have seen that summation, okay? Temporal summation, okay? So you probably have seen that somehow, okay? So the trick of the tetanus provides temporal summation, okay? To bring the voltage of the cell to sufficiently to remove the magnesium block. If that is the case, then you don't need high-frequency stimulation, okay? So if you need to bring the voltage of the cell to this level, you can either do it by high-frequency stimulation, or you can inject current with an electrode inside the cell to bring the whole cell to that voltage. Okay, just passing current through the recording. And that's what they did. So they record the cell in here, okay, and they see the responses, and at this point they keep the same regime of stimulation. But what they did, they passed current, positive current through the electrode to make the cell very depolarized. So now the magnesium that was blocking all of those NMDA receptors was not blocking anything, and then the NMDA receptor was happy and opening fully, and in that case, calcium was in, and that allowed LTP to happen. Okay, so you don't need high-frequency stimulation. The only thing that you need is the cell to be sufficiently depolarized. Okay, because then you stop the depolarization, you go back to the initial regime, and actually you realize that the cell is, those synapses potentiated. Okay, let me see. And of course, if during the tetanus you pass a negative current to bring the whole cell to negative, you block LTP. Okay, and basically, of course, if you block activation of NMDA receptor with drugs, you also do it. Okay. So, and LTP, so basically what you need is to activate the NMDA receptors, and the NMDA receptor by itself, they don't do anything. It's just they let calcium go in, and that calcium going inside the cell is the important thing. How you can show that? Because if you block the increase of calcium, you can put in there a calcium chelator. You know what a calcium chelator is? Yeah? You can fill it up the cell with a calcium. The calcium chelator is basically a molecule with two arms that will bind the calcium, basically. So if the calcium is free, it's no longer free, and therefore there's no more intracellular calcium. And you fill it up the cell with that, and you basically you don't have any more LTP. Okay, so you need calcium. So basically, recapitulating now, the tetanus is a way of depolarizing the cell. And you depolarize the cell just to fully activate the NMDA receptor. And you activate the NMDA receptor just to put calcium in. So if you put the calcium in by all the means, you bypass all of that. Okay? That's the whole idea. And so what calcium does inside, well, we don't know. Okay? One idea, so this finished by 2:20, isn't it? So, sorry, you will never know what calcium does. Okay, don't worry, because there are many enzymes candidates, and this is one of them. But I'm not gonna tell you more than that. And I will not interrogate you about that. There are many enzymes that can sense the changes in calcium and can act upon. And they're still controversial. Every year, people discover new enzymes, new kinases, candidates. So probably we have about 20 now. The one that they showed in here is the most popular, but it not means it's the only one. So why bother? Okay. But now is the question, and this is an interesting thing, the question of expression. So we know that we need coincidence of presynaptic and postsynaptic activity to induce LTP. Okay, through the activation of NMDA receptor. But what it change? How the synapse get bigger? And there are two ways, or conceptually you can divide the turf in two big areas: presynaptic changes and postsynaptic changes. I mean, and I'm going to go through a few of them just to. So presynaptically, you can increase the probability of release. You remember that quantal release in synapses, okay? So if you increase the probability of release, more likely you're going to get a response. Basically, it's kind of obvious, okay? Or you can put in each one of the vesicles, you can pack more neurotransmitters, so you have more neurotransmitter. Basically, you will have more glutamate. You can activate more AMPA receptors. Okay, so basically, the presynaptic can talk louder, so to speak. Okay, or you can make the postsynaptic to listen better. So you can put more AMPA receptors. So the same vesicle, the same glutamate, but you have more AMPA receptors in there, you will get a bigger response. Or you can jack up the response of each AMPA receptor. You can increase the conductance, the unitary conductance, for instance. Okay, so the presynaptic and postsynaptic possibilities. And I bring this because in the 90s, that was the big issue in neuroscience, whether LTP is presynaptic or postsynaptic, whether memories are encoded by changing the presynaptic release or are encoding by changing the postsynaptic response. And that was a big issue, and there were careers made and big fights, and people hate each other, and it was fun to watch all of that. Okay, and also there was some interesting twist on the story, and that's what I'm gonna go, because history and neuroscience kind of go together. So for a while, the postsynaptic camp was winning. Okay, so they made some experiment that can only be interpreted, they say, as postsynaptic changes, and they were boasting, you know, and they were very, we won, until the presynaptic camp came with this very neat, very elegant experiment that ruined the postsynaptic camp. So remember that synaptic release is a stochastic, have a stochastic nature. So sometimes you release, sometimes you don't. And here, what you have is in slices of very young animals, you can stimulate a single axon and record from the cell, and basically you're stimulating a single synapse. Okay, very delicate experiment. And what you record is that sometimes you don't get nothing, a failure, okay, because probability of release, sometimes you have probability of no release. Sometimes you release one, sometimes you release 2, 3, or 4 vesicles, okay, because that's the nature of the thing. So you have a number of release sites, all of them independent. Sometimes you release one, sometimes all of them, sometimes nothing, okay. And then that's before. And what happened after you do the tetanus or the pairing or something to induce LTP? Here's the picture. Now responses are generally bigger. Okay, so you have something like you are releasing several quantas in here more than here, and clearly you have less failures. Okay, so if you reduce the probability of failure, it goes without saying that you increase the probability of release. I mean, it's just no other interpretation. Okay, so that was a brutal demonstration that after inducing long-term potentiation, the probability of failure is reduced. Therefore, long-term potentiation is a presynaptic origin, basically undeniable thing. Okay, and so much for the postsynaptic camp. But they didn't count when there was this very smart woman. I forgot her name. That is very. And her name is very funny because her name is Francis, so I thought it was a guy, just to show how bigotry is inserted in the guy. She's very smart, and she say, wait a minute, just because you see a lot of failures, it doesn't mean that it's a presynaptic failure. Okay? Francis Edwards. Okay, that's it. And she say, just because you see a failure, it doesn't mean that presynaptic failure. It can be a postsynaptic failure. So she envisioned a little bit more sophisticated scenario, but more interesting too. So they say, suppose that synapses, when they're formed, when they're born, contain only NMDA receptors. Okay, these green things are only NMDA receptors. And through a process of maturation, okay, involving LTP, then these synapses acquire AMPA receptors. Okay, so now what will happen? So if you're recording from this synapses, which normally people do in voltage clamp, but they're very negative potentials, you will not see the NMDA response because the NMDAs are blocked by magnesium. So if you record and stimulating here, recording here, you're not going to see nothing. You're going to see there is no synapse in there, or there's a lot of failures. Okay, but now if the process of LTP inserted AMPA receptors, those are not blocked by magnesium, and then you can see them. Okay, so you will see a lot of failures, and you will see less failures, but that was actually a postsynaptic failure. All right, so and that so basically this is a silent. This called now a silent synapse. Okay, and that's actually the current model of how LTP happened. And again, like Hebb, Francis Edwards figured the thing, you know, like quite a few years before people demonstrate that. And the demonstration came slowly, and here's one of the demonstrations done here at Hopkins. These are all the results. Here what they did is they stained synapses, okay, with antibodies against NMDA receptors and against AMPA receptors. And the whole idea is that sometimes they coincide, okay, but sometimes they don't. Sometimes you have in here some red spot that means synapses that only contain NMDA receptors, okay? So synapse can contain NMDA receptors or AMPA and NMDA, okay? And okay, we are getting there. This is another interesting and beautiful early experiment, classical now, in which they use two-photon imaging. Are you familiar with the two-photon? Yeah. Okay, so this is one of the, actually, I think, one of the first utilization of the two-photon for in synapses. Okay, so what they did in this experiment, these are very old, so the quality of the signal is not that nice as can be now. But what they have here are culture cells in which they overexpress AMPA receptors, and the AMPA receptor was tagged with the green fluorescent protein, so they was fluorescent. Okay. So here you have a dendrite, okay, and another magnification, and then they make the tetanus. And what you're observing here is that this is before and this is after. You can see that this is actually that's the spine. You can see a lot of fluorescence in the spine. So all of the AMPA receptors were confined into the dendrite. You induce tetanus, and many of those AMPA receptors move from the dendrite into the spine, into the synapse. Okay, so you have kind of like a translocation of. All right. So and that's showing that this is what happened during the tetanus. Okay. This is a little bit more complicated, and this is a demonstration with electrophysiological techniques, with recordings. So bear with me. So what you're doing here, you're recording from a cell with an electrode and maintaining the membrane at minus 60 millivolts. Okay. Now this is a silent synapse. Okay, so at minus 60, the NMDA receptors are not cannot activate because they are blocked by the magnesium. So you don't see any response. So you see kind of like flat in here. Okay. Then you move to plus 30, very depolarized, no magnesium, and then you can get a response. Okay. So in this case, showing that happened basically. This is how a silent synapse will look. Now, you take one of those silent synapses, because you detect it, and then you say, can make it unsilent now. Can I put AMPA receptors? So you go to the next stage, in which you go to the minus 60, but in this moment here, you do the pairing. So basically, you depolarize the cell, and you activate a lot of that, trying to activate as much as you can the NMDA receptors to induce LTP. As a result, now you're going to get AMPA receptors inserted into that synapse. And then you come back, and now you can see responses. These are the responses that you see. So basically, you have zero response before, and out of the blue, now you get response. Okay? So a synapse that was silent, because the synapse existed in there, and now have AMPA receptors, basically. And now, of course, then you can do more LTP and add more AMPA receptors later. Okay? So this is the current view, is that actually you have the NMDA receptors allowed to conduct calcium, and somehow through this kinases that I didn't show you, it will activate processes in here which are not fully understood, and will allow lateral diffusion of AMPA receptors that are sitting outside of the synapse to move inside the synapse. Okay, and that will increase. That's basically LTP. It's lateral diffusion. So this AMPA receptor in here was kind of silent, and you move in here. Now it can respond to glutamate. So I have 10 minutes. I'm going to talk about long-term depression, which is basically the opposite of long-term potentiation. And you need, for many years, I think LTP, long-term potentiation, was discovered or demonstrated in 1973. Long-term depression was demonstrated in 1990, so 20 years after. People started to know that you need both, okay? Because if memories are coded only by potentiated synapses, eventually, anytime you put more AMPA receptors, the spine, the synapse, have to grow a little bit to make a space for this new. So if that's the only way that you are acquiring new memories, well, after 30 years of learning your head, it will be like this, okay? So you cannot just have long-term potentiation as a mechanism. You need also long-term depression. And the idea now is that any time that you learn something, some synapses get stronger, but some other ones get weaker, okay? So it's a balance, I think. Okay, or like for instance, you can learn that the rat can learn that you have to go to the left to get the water, and then you can teach the rat later, no, don't go to the left, go to the right. Yeah, this is left, this is right. Yeah, and then basically what you're telling the rat is forget about that, learn about this. So do depression for this pathway and do potentiation for this pathway, and basically you ended up the net. If you add all of the synapses, you ended up zero. Okay. And so you need a mechanism for depression. And people were looking for that. They couldn't find it. And actually, I was a postdoc in this lab when they found it. It was totally serendipity. So it was this graduate student. She was looking for long-term depression, and I was trying different protocols. Okay. So and the basic idea was, well, if you need a short, brief episode of high-frequency stimulation, like 100 Hz for a few seconds, maybe for doing a depression, you need low frequency. Yeah, but how long? Well, maybe like a few seconds or something like that. So she was doing low frequency, like 1 hertz, for a few seconds, like a minute or something like that. And nothing was going on until one day, she turned on the protocol, and then she remembered that there was the pizza lunch. Anytime that we have a speaker, we brought them to have pizza with the students. So she went to the pizza and get engaged in the conversation, and then she remembered, oh crap, my experiment. So she went back, and 20 minutes had passed, you know, and then she stopped at 1 hertz stimulation, and resumed the baseline, and then the thing was totally depressed. At the beginning, she thought it was dead, but actually recovered a little bit, and it was depression. So actually, she realized that in order to do long-term depression in a reliable manner, she had to stimulate a low frequency, but enough time to eat a pizza, which is about 15 minutes. Okay, so that became the standard, and now this is the standard model for inducing long-term depression, 1 hertz for 15 minutes. It doesn't mean that that's the way that happened in the real nature, but this is the convenient way that happened in the lab. Okay, so you can induce LTD, you know, by prolonged low-frequency stimulation. And the first question that people say, okay, so now if you need NMDA receptors to induce long-term potentiation, well, if I killed the NMDA receptors, I should induce no long-term potentiation and should get the mother of all long-term depression, okay? Just to make things better. And they tried, they put a blocker of NMDA receptors, APV, and surprisingly, it blocked LTD. And that was very puzzling and very annoying because, hey, how come that the same NMDA receptor is good for potentiating synapses and the same NMDA receptor is good for depressing synapses? It just do everything, okay? So it's kind of like a, what is the, and also they tried the calcium and it was the same. You need NMDA receptor activation and you need calcium to go in to induce LTD, just the same way of a long-term potentiation. So how come the same signal can go both ways? And here is kind of like the, not the answer, I would say the rationalization that we have now. Okay, so it's not the nature of the signal, but the shape of the signal, what people think. So what happened is that this is the NMDA receptor, and during high frequency stimulation, basically what you get is a very brief and very high calcium signal, okay, a spike. And that spike, the idea, it will activate kinases, this chem kinase 2. And that, in turn, it will be responsible somehow for LTP. Whereas if you have a low frequency stimulation, okay, you don't get that much calcium signal. It's kind of small and sustained, and that will activate preferentially the opposite of the kinases, phosphatases. Okay, that they will tend to take the phosphate from the substrate, and that actually it will be conducive to LTD. So you can imagine like a target protein, nobody knows what. If you phosphorylate, it will be good for LTP. If you dephosphorylate, it will be good for LTD, and you activate the phosphatase better with low calcium. Okay. As you increase the amount of calcium, then you start to recruit kinases, and the kinases beat the crap out of the phosphatase, basically. And then that's the kind of like the current idea. So you have a biphasic role of calcium and NMDA receptors. Low NMDA receptors, low calcium favors LTD. High calcium favors LTP. And this is kind of like a statement, kind of a disclaimer, that to the many others researchers on the area, that there are many other forms of LTP and LTD. It's just that the one that depends on NMDA receptors is the most popular and the more interesting, because it fulfills the Hebbian rule and the anti-Hebbian rule, and conceptually is easy to work with. So you will see a lot of also a lot of theoretical work done on the NMDA dependent form of LTP and LTD.
Did you say the low calcium was associated with the LTD or LTP?
LTD. So low calcium activate preferentially phosphatase, and those will tend to remove phosphate from some yet to be established proteins, okay? And that will be the signal for LTD, for depression. When as you increase the calcium now, you may activate kinases, and kinases will be, the activity of the kinase will be bigger than the activity of the phosphatase. So in the balance of phosphatase and kinases, the kinases will win, and you will have a net phosphorylation, and the net phosphorylation, it will be conductive to LTP potentiation. Okay, that is the way that people envision those things. Okay, that's the best that we can come up. All right, so yeah. Oh. So in 1 minute, I will just give you an idea of this, okay? So because I don't have the time. Yeah, I think I spent too much time in other things. So the big question is, are LTP and LTD involved really in learning a memory? So how you can prove that? And I'm gonna not go into the details, but there are 3 strategies have been tried. One is pharmacologically. So you can block the activation of NMDA receptor. You can make a hole and put the drugs into the ventricle, okay? In this case, APV is the blocker of NMDA receptor that normally block LTP and block LTD, and you do that, and then you don't get any LTP, and you don't get any new memory formation. And this is done in the Morris maze. You're probably familiar with the Morris maze. Yes? Yeah. Anybody knows? Yeah? Yeah? No? Okay, so the Morris maze is kind of cool, actually. It's a round tank, okay, with the water, and there's a submerged platform in there. So the rat or the mouse have to learn where the platform is, and that's the memory, okay? And the only clue that they will have is kind of like external clues, special clues outside. And the water is opaque. Okay, at the beginning they did it with milk. That was a bad idea because after a week, you know, rotten milk and mouse poo, it was out. But now they do it in other ways. But you drop the rat in there, and then the guy had to find the thing, and big motivation. And then once the guy learned the location, you remove the platform, okay, and you see how the animal is looking for the location. So you can see, oh, the guy really remembered the location because he's swimming desperately where the platform was, okay? Now, if you block the NMDA receptors while the guy is learning, it will just look all over the places. It never remember, okay? That's the idea. And you can do genetic manipulations. You can eliminate now the NMDA receptor from the hippocampus, and the guy never learned, you know, what it was. The other one that was kind of cool and very is saturation of LTP. So if you get LTPs in some pathways and not in the other ones, and that's the basically learning. If you go with some electrodes and saturate LTP in the hippocampus, you should eliminate the memory, because it's basically some synapses is stronger than other ones. That's basically memory. So if you make all of them like this, you don't have any memory. It's like if you go to your computer and you initialize to once all of your hard drive. You put ones everywhere, you eliminate the memories, okay? So you do that, actually, you can also eliminate the memory of the platform. To do that is kind of nasty. What they had to do, it was to remove one hippocampus of one side, one of the bananas, okay? And then the rest of the banana, they burn it until you get like a third of the banana, very small, you know? And then they put an electrode in there, and then when the guy learned a little bit, then they blasted and do the tetanus on them. And okay, so now I am 5 minutes over, so I think that's it, okay?